Ladies and gentlemen, I present to you Mr. Seekers. Thank you very much for that warm introduction. Appreciate that. So we are back now once again, going back talking about Father Gregor Mendel and his discoveries from the pea plant experiments that he did. So where we left off last time, we were talking about the fact that he had discovered this ratio, this 3-to-1 ratio that kept popping up in all the subsequent F2 generations from his studies. So it didn't depend which particular trait he was looking at. He would see that there would exist this 3-to-1 ratio for that trait. So as an example, for the pea shape, he found that 75% of the time the peas would be round, 25% of the time they would be wrinkled. For flower color, 75% of the time they were purple, 25% of the time they were white. And then something like even plant height, 75% of the plants were tall, 25% of the plants were short. So the 3-to-1 ratio continued to hold no matter which trait it was he was examining. So from this data, he was able to draw a few conclusions. He actually formulated 2 laws. So the first one he called the law of independent assortment of alleles. We'll get into what an allele is a little bit later. Just for right now, note that what he was discovering was the fact that these traits are inherited as discrete units. Today we call them genes. In his time, he called them factors. He certainly did not have any knowledge of DNA or chromosomes. The word gene, like I said before, didn't even exist yet. So he just called them factors. And so if we were looking at one particular trait, if we were looking at this purple flower color trait, for example, what he would note is the fact that this particular trait existed on a particular chromosome. And what he was saying was the fact that even though a plant exhibited purple flowers, that did not mean that it must also exhibit perhaps tall height or wrinkled seeds. So each trait was independent. So it's law of independent assortment alleles. Each trait was independent of any other trait on that chromosome. So it also held, of course, for white flowers, same sort of thing. On this chromosome, we have a particular allele coding for white flowers. Just because something has white flowers, however, does not mean it's going to be short. It does not mean that it's going to have wrinkled or round seeds. All these traits are independent of one another. Something else he discovered was this law of segregation. And what he was saying was that every living organism, of course, contains DNA. We know that. But every living organism, whether it's a human being or an avocado or a whale, doesn't matter what it is, it inherits 2 copies of each gene, 1 copy from mom and 1 copy from dad. So one thing you might be thinking about is maybe when we haven't talked about mitosis yet, but when we do talk about mitosis, you're going to discover that mitosis occurs when a cell makes an exact copy of itself. So cells do, of course, duplicate themselves, reproduce over time. And what they'll do is they will make sure that each cell it produces, each daughter cell, is an exact copy of the parent cell. So it's going to have the exact same number of chromosomes because it's an exact copy. However, with inheritance, we actually have these copies separating from each other during gamete formation. So when a sperm or egg is produced, a gamete is a sex cell, so a sperm or egg is produced, it's going to contain half the genetic material. So during meiosis, which we'll get into later, we talk about how the chromosomes, which are together, will basically pull apart during gamete formation. So you end up with half genetic material from mom and half the genetic material from dad. And then finally, when fertilization occurs, when sperm meets egg, then you'll get the total package. You'll get both copies of genetic information. So he discovered this independent assortment of alleles where none of these traits have anything to do with the other ones in terms of what's being expressed. And he also discovered this segregation component, whereas we actually get 2 copies of each gene, 1 from each parent. I would advise you to perhaps make some flashcards or put together a Quizlet because this is a very vocabulary intensive unit. There's quite a bit of vocab. Some of it may be a repeat things you've heard before from previous classes, but some of it may also be new. So just to sort of try to keep it all straight, you might want to maybe make some flashcards. So when we're talking about this, though, when we talk about genes, what we're talking about is a very specific portion of DNA. We're talking about a specific portion of DNA that codes for a very specific trait, okay, a specific characteristic. It's going to code for a very specific protein, to be exactly precise. So genes code for proteins, and genes are sections of DNA that code for proteins. So every gene, no matter what it is, whether it's height, whether it's flower color, whether it's eye color, whether it's hair color, any and every gene in your body has a very specific location where it's going to be found. And that location is called its locus. It's basically the gene's address. So if something codes for eye color, for example, then it's going to be found, on the 18th chromosome, in a particular band on the 18th chromosome for every single human being alive. You won't not find the genes responsible for eye color located anywhere else than that particular locus, that very specific address. Just as I am, my address is Fairfax, Virginia. Yours might be Arlington, it might be Reston, it might be Vienna, who knows? But that is your locus. My locus is Fairfax. You're not going to find me anywhere else. That's my address. That's where I live. Genes the same way. So they're going to be found on one particular chromosome or several different chromosomes. We'll get to that later. But they're not going to be found elsewhere. And of course, these genes that they're encoding for lots of different types of proteins that produce a wide variety, a diverse range of traits. If we were actually going to take all of that genetic material, though, take every single chromosome in your body, put it all together, that's going to be known as the genome. So the human genome was actually discovered late 1999, early 2000, if I remember correctly. So we actually have mapped out our entire genome. It is known to us now. We've known it for about 20 years. But all the genetic information, all that detail is known as the genome. So on this slide here, what I'm showing are two different chromosomes, and we're actually going to refer to them as homologous chromosomes. One of these chromosomes, maybe this top red one, came from dad, and the bottom blue one maybe came from mom. But they're called homologous because homo meaning same. They carry the same exact genetic information on them. Perhaps here we have a gene for flower petal color. Maybe here this is one that's coding for maybe pea pod shape. Maybe this one here on the far right is coding for plant height. So all these genes are found on the same chromosome. The one from dad is going to be found on the same number, same type of chromosome that came from mom. All right. So maybe this is chromosome number 22 or something, or 21 or 22. It's going to contain all these different genes. The one from mom and the one from dad are going to be the same number. Okay. So they're homologous chromosomes. What they'll do is, of course, during fertilization, they're going to line up. The homologous chromosomes line up. And what they're doing is basically comparing which traits they're carrying information for. So if this far left one here, the capital letter P, is representing flower petal color, then it's going to line up, of course, with the particular chromosome from mom's side that also codes for flower petal color. And maybe this A here represents seed shape. So the seed shape for dad is going to, of course, line up with seed shape from mom. And maybe this last one here is plant height. Plant height gene for dad is going to line up with plant height gene from mom. And then they'll basically kind of like exchange trait information to determine which particular trait is going to be exhibited on their offspring. So what we're doing here is with flower petal color. If we go back and look at Gregor Mendel's chart, you can see that 75% of the time plants exhibited the trait for purple flowers. So the dominant trait, the one that's exhibited 75% of the time is called the dominant trait. That one's going to be represented by a capital letter. The recessive trait is going to be represented by a lowercase letter. So if this is capital P, we know that both mom and dad are carrying the dominant trait for flower color. And when we put those traits together, what we get is what's called a genotype. So the genotype is just a representation of which traits are present from each parent. So we see capital P, since they're carrying the same trait, capital and capital, and they're both, they're both uppercase, so that means they're both dominant. We call them homozygous. It's a homozygous genotype. Okay, so this particular offspring is going to have purple flowers. It's got the dominant purple gene from dad and the dominant purple gene from mom. Offspring will also have purple flowers. Here in the middle, let's pretend this is also for flower petal color, just to keep this going. What we would actually do is the recessive trait is always going to be the lowercase version of the dominant trait. So in this case, we see lowercase a. We actually, in reality, would change those to, this would be a p, lowercase p, and a lowercase p. Because we know over here, it's capital P for dominant and capital P for dominant. So here, since the recessive trait is being carried, we still call it homozygous because it's the same type. It's lowercase, but this time it's homozygous recessive. Okay, it's going to be homozygous recessive because it's carrying the recessive trait for both. So if it's homozygous recessive, then the recessive trait is actually exhibited. So this offspring would have white flowers. There is no purple gene on either parent, so the offspring will have white flowers. Oops, sorry. No, don't want to. No, resume, please. Thank you. Over here we have a dominant allele on one gene from dad and a recessive allele carried over from mom. So we have the dominant represented by the capital letter and the recessive by the lowercase letter. So in this case, in that case, what we would do is we would carry that as a capital P, purple flower, and a lowercase p for white flower. And this would be called heterozygous. Hetero meaning other or different. So this time we have two different traits from each parent. But the thing to take away from this is when you have a capital letter and a lowercase letter, whenever you have this heterozygous trait showing itself, the dominant trait is what's going to actually show up in the offspring. So keep in mind that dominant always masks recessive. Dominant is always going to cover up recessive. The only time we ever have recessive traits show up in offspring is when both parents are contributing the recessive trait. If one parent is contributing the dominant trait and the other parent is contributing the recessive trait, the dominant trait is going to show up. So for example, my mom has brown eyes, my dad has green eyes. Brown is dominant to green. So I got the brown eye gene from my mom, the green eye gene from my dad. I'm most likely heterozygous in that regard. But because I did get the dominant trait, the brown eye gene from my mom, I have brown eyes. So something like hair color, even something like eye color is actually controlled by a number of genes. It's not just one. So it's much more complex than this, but I was just trying to give a simple example there. I think for now, that's good. That's kind of like where we want to leave it, just the introduction to some vocabulary, what we mean by these terms. If you have any questions, please feel free to email and let me know. For now, we'll go ahead and end it. So please stay well, stay safe. Until next time, goodbye.